Welcome to Lesson 6 of Module 7 Infectious Disease. In this video, we're going to be looking at immunity and we're going to be focusing on our adaptive immune system, but also looking briefly back at our innate system and how they work together to respond to a pathogen. So the human immune system is made up of both the innate and the adaptive immune response. And our innate is what we covered in Lesson 5, which is a genetically pre-programmed response. The repeat exposure is the same response. There is no learning or memory formation in the innate immune response system. However, when we look at the adaptive immune response, these are actioned when the innate system and the innate response fails to clear any pathogen. It's also known as our third line of defense or acquired immunity. So there are two arms of our adaptive immune response system, and these are known as our humoral and cell-mediated responses. There are cells responsible for generating this response, known as our B and T cells. It is a specific response system and it involves a great diversity of possible responses. It has a memory system and it's capable of self-tolerance. So here we have our two arms, our cell-mediated and our humoral response. So on our cell-mediated here, we're seeing macrophages presenting an antigen to the lymph nodes. And then we see our helper T cells, then activating our T cells. So our cell-mediated side is really a lot to do with our T cells and how our cytotoxic T cells will then move to our infected cells, release chemicals that will destroy the cell and the pathogen within it. We then also at the same time are producing our memory T cells, which remain in the body, and if we are re-exposed to the same antigen, this produces the same response. In our humoral response, we're looking at our B cells. So we see our antigen-presenting B cells that work with the helper T cells to match an antigen. And then we see our B cells being activated, being produced, and then moving to our lymph nodes and working with our antibodies to form our antigen-antibody complex. And then we're seeing a destruction of the pathogen. At the same time here, we're also producing memory B cells. So therefore, when we're re-exposed to the same antigen again, we see a large production of our antibodies. Both our B cells and our T cells, so they're actually called our B lymphocytes and our T lymphocytes, but we're just going to refer to them as our B and T cells, are both produced in the bone marrow. However, the B cells mature in the bone marrow and the T cells mature in the thymus gland. And then we can see here that they're released into the same places. However, then after that, there are performing different functions to help our body defend itself against the pathogens that are trying to enter. So here we have that in a bit more of a word format. And we can see here that the most important point is that our T cells control cell-mediated immunity, and then our B cells control the antibody-mediated immunity. There are 4 different types of T cells which we see in our cell-mediated immunity. So we have our helper T cells, our cytotoxic or our killer T cells, memory cells, and then our suppressor T cells. So each of these work together but also perform different functions to help activate and perform that cell-mediated immunity. So our antibodies, which are obviously part of our antibody response, are produced because of the presence of an antigen in the body. And these are produced from plasma cells when our B cells are activated. This forms the antigen-antibody complex, which eventually causes the deactivation of the antigen. And then these 5 processes are undertaken in order as they're written there to destroy the pathogen. So we have the neutralization, agglutination, precipitation of dissolved antigens, activation of the complement system, and then our opsonization. So here we have just a few pictures here which talk about our antigen and antibody complexes and how they will result in us performing our immune responses here. So our antibodies have antigen binding sites, and you can see here that the antigen binding sites produced from each antibody are specific to each antigen. So we do have to produce antibodies specific to each of the antigens that are present on each different pathogen, which is why we have different responses and different vaccinations and different ways of treating different infectious diseases. Memory cells are produced to eliminate an antigen rapidly the next time it appears, which allows any of our symptoms to not appear and for them to not take effect on the body. So on the graph here on the right, we see our primary immune response. So this is when we're first infected and we see this antigen. We produce a smaller amount of antibodies, however, than we do the second time. So we see a faster, a larger, and a longer response if we're produced a second time. So in the primary immune response, we see our antibody-producing plasma cells and cytotoxic T cells being produced and stored for the next encounter. And then with our secondary immune response, there is a more rapid response because the production of the antibodies is much greater here. Some immunity might last a short time and others for the rest of a person's life. If they do last for a short time, that's where we're seeing people receiving booster shots. So here we have a whole picture of how the T cells and the B cells both work for antigens and resulting in obviously a person no longer being affected and having these memory cells in place to have a better secondary response. So this is just for your reference later, and we can see how all the different types of T cells and B cells play a role in each of these different steps along the way. Again, another kind of diagram here. So we're seeing so there are both memory cells being produced for T and B cells, but both are then later activated in our secondary immune response. So this is just a small bit of information about dengue fever and the responses that the body will take place if someone is infected with dengue fever. So the virus is transmitted by the mosquito, and some of the symptoms a person might receive are fever, headaches, rash, vomiting, and muscle pain. The target is the innate immune system, and the virus affects the macrophages and the dendritic cells and can become bloodborne once it evades the innate immune system. Interferons are not produced, and therefore they're unable to signal any helper T cells to become activated, and then it can form intracellular membrane pockets. And then when we're trying to look for dengue fever, we see a change in our blood count. So there's a reduced white cell count, reduced number of platelets, and the percentage of the red blood cells in the blood increase due to dengue fever in a person's body. So this is how the humoral immune system, so the adaptive, the specific immune system, is affected in dengue fever. Thank you for tuning in for lesson 6. Make sure you tune in for lesson 7.